And that leads us to stage 3. Cell-like structures are formed and the polymers become enclosed. We need to surround the polymers with a membrane. We have to completely encase them. Now, we've heard of some membranes already. What are the membranes that you guys know of made out of? What are cell membranes made of? I hoped we knew. Yes? They're both hydrophobic and hydrophilic at the same time, so they're termed amphipathic. Now, do you know an example of an amphipathic molecule? Looks sort of, it's got this polar head, 2 nonpolar tails. You guys have heard of phospholipids before? Yeah, phospholipids. You've extensively studied phospholipids, I hope, to where you know the head orients toward water, the tail orients away. That's one way of forming a membrane, but there are other ways as well. What we need to do is we're going to create these things called protobionts. They're not really alive yet. They don't count as life. They're not multiplying. Protobiont is a bunch of prebiotically produced molecules that got a phospholipid bilayer or a coacervate layer or some sort of layer to them so that they have an inside and an outside. As you can imagine, it's very important for living organisms to have both an inside and an outside, an internal environment to regulate and an external environment to react to. This is probably the most common one that we know of today, the lipid bilayer. You've got phospholipids with their polar heads and their nonpolar tails. So they're hydrophilic heads, they're hydrophobic tails, making them amphipathic. The heads orient toward water over here. The tails orient toward a space with no water. And then you've got another side with the heads facing outward toward an external aqueous environment. We need a boundary. We need that inside and outside. Once the polymers are bound, you may have very, very simple metabolic pathways. The next step that has to happen is enzymatic reactions. You need to build catalysts. You need to, in some way, these molecules need to be able to build themselves. It can't just be a random event. It has to be able to make it so that it's easier and easier and easier to bind these molecules together. If this crucial event occurs, if you're able to have an enzyme or something that speeds up the reaction, suddenly that protobiont can begin to grow. Because now it's building things inside of itself that it'll use to build more of itself. It gets bigger and bigger and bigger. So we have to get that enzyme. We have to get that enzymatic linking ability. Once you get that ability, the ability to reproduce the internal parts, to reproduce all of the components, allowing it to grow, they were able to split. Because you see, when you've got these structures, they can only grow to a certain size. You can't keep growing bigger and bigger and bigger. You can't have a unicellular organism that's as big as a dog, because the membrane won't hold itself together. It's almost like, you guys have blown bubbles before, right? Bubbles have a maximum size. You can't just keep blowing air into them and they will get bigger and bigger and bigger. What happens to them? They pop. Luckily, these cells don't pop. What happens is they get bigger and bigger and bigger, and then the center ends up pinching off. And you have 2 cells then. Thing is, once they split like that, each side is going to get some of that enzyme and is able to reproduce macromolecules. So if they're able to reproduce, if they're able to enzymatically link molecules, it's not true reproduction yet, you end up with a protobiont that's able to grow bigger and split, grow bigger and split, grow bigger and split. It's starting to seem more and more like life. Now there's 2 possible protective coverings these may have had? Liposomes or coacervates? Liposomes are a little bit more biologically complex. Coacervates, pretty simple. Liposomes, you know. Liposomes are phospholipid bilayers. You may also have these coacervates, a protective covering. If you were to take some strings of proteins, some polypeptides, which at this point we know are being produced in water, some carbohydrates, some nucleic acids, again, that we know are being produced. If you were to drop them in water, due to what's called the hydrophobic effect, they may end up orienting in such a way to form a barrier. Just automatically, all by themselves. It's passive. It does not require energy for this to happen. It happens automatically. It ends up creating, actually, it attracts water molecules and creates a boundary out of that water. So now you have an inside, that's an aqueous solution, a boundary, and then a slightly bound outside that's an aqueous solution. Anything that's trapped on the inside, any of these molecules trapped on the inside, start to perform primitive metabolic functions. They start to polymerize. And the other more complex one is the liposome. Liposomes are made out of phospholipids. Polar head, nonpolar tail. They'll form either liposomes, which are primitive cells, an outside, a hydrophobic region, and an aqueous inside, or micelles, where they are all the tails are facing inwards. That's sort of what happens when you wash dishes and form bubbles. With liposomes or coacervates, once you've got that membrane, you can start to form electrochemical gradients. What did we previously use electrochemical gradients for? Where have you guys experienced electrochemical gradients in biology thus far? We haven't talked about them this semester. Right, you're going through the cell membrane. You're passing through this particular protein complex called ATP synthase. So these hydrogen ions or protons stream through it. They spin it around, and that creates energy. You can use the energy of a gradient, having too many molecules on the outside or on the inside trying to stream out, to generate energy for forming new molecules, for forming bonds. This is another, once you have these membranes, now you have the potential to create energy necessary to drive life. All right, so this is kind of cool too. Back to clay, because clay is important. If you were to take slightly moist clay, I don't know, clay hanging out at the side of the ocean, it will catalyze the formation of liposomes. It builds liposomes. It makes liposomes happen much faster than they otherwise would. This is even more important if RNA is present. RNA is a nucleic acid. If RNA is present, that liposome will form around the RNA in moist clay. So, just to get us caught up to date with these three steps thus far. We're building these original molecules. We're building these pre-organic molecules. That could happen at the bottom of the ocean. That could happen through the reducing atmosphere. And it might happen with meteors crashing onto the planet. It's possible. In any case, these pre-organic molecules are sitting in the water. They're becoming polymerized. Whether they're polymerizing on clay or at the geothermal vents, they're polymerizing. They're becoming these bigger molecules. You're building RNA. If that RNA was built on the clay, or it lands on the clay, that will then, the clay will form a liposome around it, or make a liposome form around it. So now you have these early protobionts surrounded by a liposome that could potentially have an electrochemical gradient associated with it. RNA is going to be important. We're going to see RNA